Hello, we have Karan Syal with us today. Hi Karan.
Hi Olive, how are you?
I'm good, man. How's where are you right now? You in the campus?
So, I am currently in my room. I go to ASU, which is Arizona State University,
Uh,
for my PhD in biological design or biomedical engineering.
okay, so
So,
yeah,
yeah.
I know. Karan is an experienced researcher, so I'll let you give the introduction fully. So yeah,
Sure.
tell me in details like how where what have you done and like from your undergrad time and how you've moved on to the US and what you're doing currently.
Okay. So, I think I did my undergrad back in IIT Madras in 2000 from '05 to '010. Then I kind of worked for about a year in a consulting firm called ZS.
Yeah.
And then I kind of figured that probably I want to get back to research a little bit.
Huh.
I'm focused on a little bit more on the applied aspect of research.
Okay.
So I joined ASU in 2011 and currently I'm working in the Center for Biosensors and Bioelectronics at Biodesign Institute over here.
Okay.
So to explain my research in very layman term,
Mm-hmm.
uh, when you and I or everybody walks into a doctor's clinic
Huh.
these days,
Yeah.
so the doctor, while prescribing us antibiotics for bacterial infections, does not really know if the antibiotics are going to work or not.
Mm-hmm.
At best, he kind of takes an intelligent guesswork.
Okay.
So it turns out that in about 30% of the cases, that intelligent guesswork is wrong.
Okay. That.
And the closest clinical test which can tell the doctor if the antibiotic is going to work or not,
Hmm.
it takes about 3 days currently.
Okay.
And my research aims to do that test in real time within 2 hours.
Okay. That's very interesting, actually. Yep. Okay.
Right. So, I mean, in the bigger scheme of things, so I mean, a parallel thing to think about is that in 1920s,
Hmm.
the average life expectancy of human beings used to be about 60, 70 years,
Okay. Yep.
right?
Hmm.
And right now, it's about, I don't know, 80, 85 years.
Yeah. Yeah. I mean,
So,
the
the,
global average is obviously high. Yeah. So like the Japanese
right,
and the I mean even from the developed states. Yeah. So
absolutely.
yeah.
So the question is, where did the extra 20 years come from?
Hmm.
So the extra 20 years came from a single molecule called antibiotics, which the human civilization discovered by chance. So in 1920s, even if you get used to get a simple razor cut from bleeding, I mean, due to shaving and you used to have bleeding,
Hmm.
there was a chance that it might get infected and you will die.
Hmm.
So after 1920s, when we discovered antibiotics,
Hmm.
we could use these over and over again to treat any bacterial infection, and that's how we won the evolutionary war against the microbes.
Yes.
But
Yeah.
over the last 100 years,
Hmm.
we've abused these molecules so much
Hmm.
that they have stopped working. That's what we call the phenomenon of antibiotic resistance bacteria,
Hmm.
which caused over, I guess, around 700,000 deaths globally last year.
Okay.
And by 2050 is expected to cause like 10 million to 50 million deaths all over the world.
Okay.
So there is no way to diagnose these antibiotic resistance bacteria
Hmm.
clinically right now. It takes about 3 days,
Okay. Hmm.
and that's why doctors don't even know if the antibiotics which they're prescribing us will work or not.
Okay.
So in the overall scheme of things, that's where my PhD fits, that we want to give clinicians a tool
Hmm.
to test the antibiotics before they prescribe to patients so that they know if they're going to work or not,
Hmm.
or the appropriate narrow spectrum antibiotic to choose from.
Okay. So then,
Yeah,
so there are far-reaching consequences, obviously. Yeah. So that
absolutely.
that's
Yeah.
very 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 interesting, actually. So if I may ask you, so like how what kind of So tell us more about like what's your like daily routine like what kind of stuff it's how much time is spent in the lab, and how much time do you analyze stuff in your like in some numerical stuff, or what kind
So,
of stuff experiments you run, how do you do it actually?
okay. So I think let me go back into explaining a little bit that how we achieve this, all of this, and I think that will help answer, like put into place what are the different aspects of what I'm working on right now and how much time I spent on each of those.
Right.
Is
Okay.
that okay?
Yeah, makes sense. Yeah.
So I think so. What we discovered, I think I think 1.5 years back or something, like we had a vague idea.
Hmm.
So we what we did is like bacterial cells, you know,
Hmm.
we immobilized on a chip,
Hmm.
let's say,
Hmm.
and we decided to image them.
Hmm.
So what we saw is that when we look at these cells under a normal microscope,
Hmm.
these are not moving, they are very stationary. Okay?
Hmm.
But when we look under our special homemade microscope in the lab,
Hmm.
which is basically a plasmonic imaging tool, let's say, like a plasmonic microscope,
Okay. Okay.
we could see that these bacterial cells move with a very small standard deviation, like with a very small fluctuation. So these guys,
Hmm.
which to the normal microscopy eyes look fixed,
Hmm.
they're actually moving with a small amplitude of let's say 10 nanometers.
Okay.
So they're bouncing off the surface about 10 nanometers, you know.
Hmm.
So we got very curious that why exactly is this motion there for all microbial cells,
Hmm.
you know.
Hmm.
And then we hypothesized that maybe this is a signature of the metabolism of the bacterial cells.
Okay.
And that's kind of what happened. Like we like started doing experiments one by one, and we saw that when before the antibiotics, the cells are moving about 10 nanometers, and when we give it antibiotics,
Hmm.
the cells are moving about 1 nanometer. So we could get in real time the changes in
Hmm.
the motion
Hmm.
and get to know basically if the antibiotic worked or not.
So by that, if I if sorry, I'll just explain what I understood. So if
Sure, absolutely.
yeah, so if there's more deviation and then there's more motion, so does that mean it is working, or if there's less motion, that's better?
So it's kind of like the change in motion is what we are looking for.
Okay.
So I mean, initially, let's say that all the bacterial cells give you a motion in the range of 10 to 12 nanometers.
Hmm.
That means that the cell is alive. It's the same analogy
Hmm.
that when we are alive, we are moving, right? And when we are
Right.
dead, we stop moving.
Exactly. Yeah. Okay.
It's the same thing we are applying to bacterial cells.
Okay.
It's just that these cells are so small,
Hmm.
like they're on the micron scale,
Hmm.
that their motion is on the nanometer scale,
Right. Right. Okay.
right?
Hmm.
So you need tools that sensitive to measure that motion.
Yeah. Got it. Yeah.
So So that's what we basically measured. Like the scale of that motion was very small before the antibiotics, like 10 nanometers.
Hmm.
And when we gave it antibiotics, it turned to about zero point 0.5 or 1 nanometer.
Okay, so
So
so basically that's what like the bacteria then it's sort of getting killed in a way. I mean, yeah.
exactly,
Okay.
exactly, exactly. So in this, if you think about it,
Hmm.
I have one tool which is my physics-based plasmonic imaging tool, right?
Okay.
That's one aspect of this technique. The second thing is bacterial cells. That is the second aspect of this technique.
Okay.
The third thing is basically getting this signature out of the images that we record. That
Hmm.
is the third aspect,
Hmm.
right?
Hmm.
And the fourth aspect is getting data out of these images to analyze,
Okay.
right?
Hmm.
So these are four aspects in the research, okay?
Hmm.
And then if you think about it that we are claiming that it's going to be clinically useful, right?
Hmm.
So how do we know that it's going to be clinically useful unless and until we have verified with the clinicians that such a tool will work, right?
Right. Right. Of
So
course.
that kind of comes into partial, I mean not thoroughly a part of my PhD,
Hmm.
but let's say that finding out the commercial potential of this tool.
Right.
So those are kind of like the five broad aspects,
Okay.
and I think like in terms of the time I spend doing all these five,
Hmm.
I think it varies. So sometimes we call it like zoom in and zoom out in the lab. Like sometimes we have to zoom out
Hmm.
and see that where does like what I'm currently doing in terms of data analytics, instrumentation, imaging, anything,
Hmm.
where does it fit in the overall big picture?
Hmm.
And then I have to zoom in
Hmm.
and figure out the nitty gritties of how to get all of these five things working.
Right, right, okay.
So, like for example, sometime back, I
Hmm.
mean not so long back,
Hmm.
I was busy doing my experiments.
Hmm.
So I consistently did experiments for like 3 weeks non-stop.
Okay.
Okay.
Hmm.
Then I accumulated a lot of data in
Hmm.
terms of my images.
Okay.
So I mean, let's take a step back in a way. Like so, I explained the first step was my imaging tool in a way, right?
Right, right.
So I spent about let's say 2 months consecutively improving my imaging tool.
Hmm.
But if I can like get it to the higher standards that we want it to be. The
Yeah.
second part was microbiology. Then I spent about another month optimizing the conditions of the microbes.
Okay.
Then I spent the next month. So I used both of these in the last like 3 weeks consecutively to do a lot of experiments. So at
Hmm.
that time I was just doing experiments.
Hmm. Hmm.
And then I basically accumulated a lot of images.
Hmm.
And now I'm looking into the image analysis part, like how to get data out of these images. So I spent a week over there
Hmm.
just doing image analysis. Now I have a lot of data,
Hmm.
and now I'm trying to do something like PCA component analysis or something,
Okay.
like to get extract meaningful data out of these images.
Okay.
So I think in a typical day things vary,
Hmm.
and then at times I have to convince one person of the or the other about the big picture. Then
Hmm.
I go to the fifth part in that day about the business,
Hmm.
like is this commercially feasible or not?
Right.
Like what do we else need to take this out in a way?
So to summarize, what I can
So
understand is, so you need the myriad of skills. Like you need even analytic skills, like where you analyzing the data, and then you need the knowledge of microbiology when you get into
right,
the biology stuff. So, yeah,
you're right, yeah.
yeah,
It's very interdisciplinary. I think at least what I'm working on is very interdisciplinary.
okay, yeah, yeah,
And I think my motivation out of this whole thing was that I wanted to see this applied in clinics.
right,
So I think I had to figure it out like it was a natural curiosity thing that if people in clinics are going to use it or not,
right,
right?
right,
So that's when I parallelly started what we call the market opportunity or the business development
hmm,
or like business opportunities phase of my PhD in
hmm,
which I had a small student venture, like ran things, figured out the market,
hmm,
competitors,
hmm,
if the real customers in the clinics are really going to buy it or not and stuff like that.
right, right, right. So,
So
how is it like the experience? Like when student researchers reach out to the sort of the commercial market. So, how is like the response? I mean, how does it work in the US? It's very favorable, or like you have to sort of establish yourself first, and then you get a head
yeah.
start.
I mean, like, I don't know, like, what do you mean establish yourself first? Like,
As
like,
in, just give me a minute. Just give me a
right.
minute. Yeah, sure. Yeah, sorry for the interruption. I'm back. So,
No, that,
uh, but what I mean is, I mean, how open are people to, like being reached out by researchers? Like, I just want to know from a, like when young people hear about this, that they'll get excited. Like, for example, what is the scope? Like translating your own research into a, more commercial opportunity.
right. So I think it doesn't matter how, like, so, I mean, if you take a normal subset of, like, set of 100 people, they're going to be some which are going to be excited about what you do, and they're going to be some which are not excited about what you do.
Hmm.
But the bottom line is that if you claim that something is applicable in the clinical settings,
Hmm.
you have to imagine the business card of the end user.
Hmm.
What will it look like?
Right.
And you have to go talk to them. That's the only way of finding about what you're doing. Is it going to be useful or have an impact at the end of the day or not?
Right. 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 Okay.
You see what I mean?
Yeah. Yeah.
So in that, when you go and try to figure that impact out,
Hmm.
there are going to be some who are going to be enthusiastic and there are going to be some who are not.
Right.
But you will get good feedback from both the cases, right? That where do you lack and what exactly do you need to do? Like, I mean, a good example is that we I mean, like, I think we went to basically Mayo Clinic, which is a big hospital over there,
Hmm.
over here, and I spent some time in the department of the clinical microbiology,
Okay.
looking at the current assays.
Yeah.
And then the fun part was convincing everybody over there,
Hmm.
like these guys have been running these assays for about 10, I mean, like ever since, like we discovered antibiotics.
Okay.
And there are these bunch of technicians, microbiology directors, etc., who have been doing this for a very long time now.
Okay.
So the fun part is getting their feedback on what they think is going to work or not.
Right.
And it's a good exercise. So out of 10 technicians I met, maybe 4 or 5 didn't agree with me. But
Hmm.
they told me what exactly will make it work.
Hmm. Hmm.
Like somebody wants automation, somebody wants this, somebody wants that. So you get a complete picture of what people exactly want out there.
Exactly. Right.
Did that make sense? I'm sorry, I was a little vague or abstract maybe. Ask me specifics and I can share.
Definitely. No, I think what I can definitely take out of this is reaching out to people is important. You get positive feedback.
Absolutely.
I mean, you get a lot of constructive feedback, and then you can definitely work your way out of and solve whatever case that you
Yeah,
want to solve. Yeah. So
absolutely.
yeah. Now I just getting back to the like, so what exactly is the field called? And so I mean,
Yeah.
the where you're working. So is it microbiology, or I mean, it's immunology, or what kind of stuff is it?
So, I mean, you can put it in the broad category of diagnostics,
Okay.
like biosensor for diagnostics, infectious disease diagnostics in general.
Okay, so when you say biosensors, so then those
Hmm.
okay, so basically the measuring, so basically it's related to that antibiotics and reacting with the bacterial cells, and okay, so
Right, right,
okay.
right. So, if you think about it, my PhD probably has a flavor of physics.
Hmm.
It has a little bit of flavor of instrumentation. It has a little bit of flavor of microbiology,
Hmm.
then image processing, then data analytics,
Okay.
right? So it falls probably within like these 5, 6 different fields,
Okay. And
you know.
right. So do you collaborate with other researchers, or how, or do you have a team? How does it work, or you are more on the individual side as of now?
So I think like right now, I think my lab. So I think like I got this idea about I think like 1.5, 2 years back, and I
Hmm.
think my professor, Dr. Rao, was very kind to let me pursue this.
Hmm.
And I think like when I first established the whole thing, I think I was pretty like it was more or less an individual effort.
Hmm.
And then after a while, I mean there are different expertise available in the lab, and you go seek them out.
Right.
And then gradually now we have now, yeah, I mean we have collaborators. Like for example, in clinical microbiology, we consult with Mayo Clinic, which is a leading hospital over here.
Okay.
There is another microbiologist on my whom I pretty much talk to on a weekly basis. Who's on my committee as well.
Hmm.
So yeah, I mean we have different expertises available, which we go and seek out whenever we need their help.
Right. Right.
You know.
Okay.
And then there was somebody else helping me with the image analysis.
Hmm.
So I think again you can't do everything on your own, and look, like I never came with a microbiology expertise, so
Okay.
I have to fall back on somebody else's expertise who's been in this field for 30 years to
Okay.
see how does it benefit my research at that point.
Of course. Of course.
Yeah.
So okay, that makes sense. So, and what about, so I mean, you had a graduate education in biotech. So
Right.
how was that useful? As I mean, what were the things which were like really helpful, and what are the developmental areas which you think maybe if they were like there at that point of time, it would have been very useful.
Yeah, I think in general, I think like I mean honestly, I think biotech was helpful.
Hmm.
But what I really enjoyed during my undergraduate was that we did a lot of subjects.
Hmm.
We did a lot of subjects not only from bio, but we also did a lot of subjects from other engineering fields and even management sciences for that matter. You
Hmm.
know?
Okay.
So I think I'm the kind of person who kind of likes this broad, like discipline,
Hmm.
knowledge gathering, and then applying it to whatever I'm doing on a daily basis.
Mm-hmm, mm-hmm.
So that's what I kind of liked.
Okay, okay.
Did I
Got.
answer you?
Yeah, I mean,
Like,
yeah, I got it, but I mean, from more from the technical side, my question was a little more technical in nature. That
okay, ask, yeah, yeah.
yeah, more from the
Technical side, Arjun?
just core biotech. I mean, like now that you are into biomedical sort of, so
Uh-huh.
like
I
how
think
the technicalities, like how are the technicalities like?
I think it was helpful, Olive. I mean, if you think about it, I mean, I didn't, I probably got a flavor of all these things that I talked about
Hmm.
in my research, in
Hmm.
my undergraduate courses.
Right,
But at that time, I never got an opportunity to apply these,
right,
you know.
yeah,
Like, I was busy applying, so I had, like, a little bit of background and everything, but I wasn't an expert in any of these.
right.
Well, like, that's how we all are in undergrad. I mean,
Of course, yeah, yeah.
and I think probably in my undergrad, I think I was interested in solving some other problem.
Mm-hmm.
So my whole focus was not on all these subjects, but
Hmm.
it was on something else at that point.
Okay, yeah,
Like for example, I was interested in protein folding problem.
okay,
So the only two courses I really, really put my head to was two protein folding courses that I did in my 5 years.
okay, okay.
But I had the overall, like the rest of the courses with me where I didn't do so well, but okay-ish
Hmm.
as a background. So when I came over here, I couldn't work on the protein folding problem anymore
Hmm.
due to some reasons.
Hmm.
But all of these courses came in handy to like take the next few steps in finding a good project,
Right.
you know?
Yeah, I got it. Yeah, yeah.
Yeah.
Okay, that's very interesting. So, what are the like? I mean, of course, I mean, you are addressing a very specific problem, so to
Mhm.
say, and but if you can, I mean, of course, like you might have other researchers in the lab. Like, so what are the like the cutting edge things which you guys are working on nowadays in your lab? And I mean, the people you interact, you must be interacting with a lot of researchers and professors and microbiologists and
Right.
even yeah. So what kind of people cutting edge stuff are people working on nowadays in your domain, sort of?
So I think in my domain right now, okay, let me narrow it down a little bit.
Yeah. Okay.
Uh, I think in my so I mean so in infectious diseases in general,
Hmm.
there are three problems to solve. I think broadly.
Okay.
Okay. One is finding out if it's a viral or a bacterial infection.
Okay.
Okay.
Hmm.
Second problem is that if we have identified that it's a bacterial infection, what bacteria it is.
Okay.
Okay. Like what its specific organism it is.
Right.
Then the third problem is basically uh, figuring out if the drugs you're giving to these bacteria are going to work or not.
Okay.
So these are the three broad problems,
Hmm.
and I think people are trying to solve these problems from different ways possible. So my project, for example, if you could, like it basically falls into the third category right now,
Exactly. Exactly. Yeah.
right?
Yeah.
And then I definitely know of startups or other researchers which are trying to solve the second problem,
Hmm.
which is identifying like different species of bacteria in urine, sputum, this,
Hmm.
that, etc., or your blood through one way or the other.
Okay.
So I think one of the good like researchers in ASU, he has kind of like a small array
Hmm.
under which you basically give blood, like a drop of blood.
Hmm.
And the hypothesis is that there are antibodies generated against any of the bugs which infect your body.
Right.
And these antibodies will go stick and give like a signature out,
Okay.
like on that chip. Like,
Hmm.
so it's a 10,000 bell chip. So you have like 10,000 data points over there in a particular signature.
Okay.
So there are different people working with, like in infectious diseases, solving these three problems by one way or the other.
Yeah.
And if you think about the second way, like now I also made a startup some time back. We're trying to solve the second problem,
Hmm.
which is basically diagnosing urine infections
Okay.
at home or something like that.
So,
So,
just correct me if I'm wrong. So,
so
sorry, just have a question for you. Yeah.
yeah.
So, right now, as in, I mean, in the current state of things, I mean, what are the challenges really? Because I mean, currently also when we go for a routine diagnostic to a doctor, so what I can understand is so they don't really check for what virus or bacteria I have, right? So
Right.
they just prescribe me some kind of an antibiotic. So
Absolutely.
so the problem which you guys are trying to identify is actually, or maybe the other research groups which you're talking about, so is actually being able to identify the virus and the bacteria. So and
Yes.
then the kind of medication which you will give will vary with that.
Right. Yes.
Am I right?
Yes, you're absolutely right.
So
So, I mean, if you talk about challenges, there are like all scores of challenges. I mean, like like the very first challenge is that how do you get like these virus or bacteria out of a body fluid? Like that's challenge one.
okay.
You know.
Hmm.
Then the second part of challenge is that are so most of these companies right now, I mean, are relying on some kind of PCR analysis. You know,
Hmm.
like or some kind of genomic testing.
Okay.
How specific are your probes to all of these?
Hmm.
And then you have to take into account that viruses evolve every summer, every winter. They're evolving.
Hmm.
Like there's a mutation which makes the previous probes
Hmm.
outdated, so they're not able to recognize that,
Okay.
right?
Yeah.
So there are like tons of challenges, and I think like everybody, like there are different researchers working on different parts of the whole thing.
Hmm.
With and like for example, in my research, the challenge was that like so if you think about it, in antibiotic susceptibility testing, like the broader field I'm talking about,
Hmm.
why does it take 3 days for the doctors to figure out if the bug is working or not, if the antibiotic is going to work or not, right?
Hmm.
So the tools that they use
Hmm.
basically depend upon culturing the bacteria for 2 or 3 days. Like the first step is taking it out of urine.
Okay.
That takes 1 day.
Okay.
Then the second step is like further growing these cells for another 1 day
Hmm.
and checking for their growth if they're going to work or not,
Okay.
right?
Hmm.
So I mean, these are so now if you think about it, that why do you need to culture? Why isn't there a direct way
Hmm.
without culturing bacteria or growing them to figure out if the antibiotic has worked or not,
Okay. Yeah.
right?
Hmm.
So that is the approach, for example, we are taking right now.
Yeah.
And then there are other people who are focused on like so the traditional instruments you have to grow one bacteria to let's say a million
Hmm.
to figure out if the antibiotic worked or not. So the other groups are trying to image this bacterial cells
Hmm.
and get faster growth, like
Hmm.
the growth faster in a way.
Yeah.
So
Okay.
there are different approaches people are taking.
Hmm.
But yeah, these are the three broad things happening right now, and each of them has their own set of challenges, like separating bacteria from blood in the first part, or the viral particles. And I mean, sorry, I'm being a little vague, but I hope you get it.
Yeah, yeah, no, definitely. I mean, of course, I
Right,
think, it's good. I mean, I definitely, it's a very eye-opening thing for me. So, for example,
right,
to,
and
yeah, I'm
right,
sure.
absolutely. And in terms of where my lab works
Hmm.
in broader fields, so I think my lab is primarily focused on small molecule cell interaction.
Okay.
Okay.
Ah,
So to say that, like all the pharmaceutical companies, they have these drug molecules, right?
right.
So these drug molecules are very small,
Hmm. Yeah.
and there are no good assays to figure out how the small molecule interacts with the cell.
Hmm.
Okay. So my lab heavily focuses on finding new ways how these small molecules interact with the cells.
Hmm.
You know. So we basically develop the biosensor tools for these kind of like for pharmaceutical applications in a way.
Okay. Okay.
You see what I mean? So that's the broader area my lab falls into.
Right.
And we are trying different approaches to figure out if the small molecule is actually interacting with the cell or not.
Yeah. Okay.
You know?
Yeah, makes sense. All right,
And
that's like,
yeah, yeah.
yeah, that's
All
like
right.
very sort of eye-opening, and
I don't know. Yeah, it
very
was eye opening, but I definitely ended up confusing you, I'm sure.
no, that's I was open to that, learning new things while I talk with
Okay.
you. So of course it's a new area for me. So I'm sure, but I think overall, probably, I mean, you gave a very clear sort of three very distinct problem areas, and I think even if when I share this thing with a wider audience probably, and
Yeah,
they definitely relate to it, and the way you classified the problem will be like pretty useful for anybody who's interested in this kind of stuff.
yeah,
And
absolutely,
yeah, and antibiotics, and I mean, even people who are not involved in direct research, I mean, everyone gets to hear about the antibiotic resistance and
actually,
the
yeah,
challenges that people are facing right now. So it's
absolutely.
really great to speak to somebody who is actually working on that. So
Yeah, it's a pleasure. I mean, I'm trying. I don't know how far we will get with it, but at least we are trying.
yeah, of course. So probably for today, I'll keep this short, and we might come back to you again and discuss in more detail the kind
Absolutely,
of progress which you guys are making, and for if we might have some more specific questions for you next time.
will be
So
my pleasure.
it's a
So,
pleasure talking to you, then, Karan. I'll
thank you,
arrange
Alan.
another session where we probably get into further details.
Absolutely, absolutely. Thank you, Alan.
And thanks for your time. I'll ta for now. Bye.
Sure, thanks.